in retrospect, was a very ill-judged decision to put Nature onto CD-ROM rather than creating a website. And I remember one of my colleagues at another competing publisher laughing at us. I haven't followed it close enough to have a very informed opinion on that, but the one thing that will make that will cause change at places like Elsevier is I think one broad problem with translational neuroscience is, I can remember it quite vividly. It was the Slumimdo Shostakovich's Fifth Symphony. And I remember just sort of, in rehearsal, and I just suddenly decided, okay, I'm going to go to Harvard.
Welcome to Stimulating Brains.
Hello and welcome to Stimulating Brains. It's Ala again. For this episode, we had the pleasure of talking to Dr. Charles Jennings, who is the Executive Director of the Program for Interdisciplinary Neuroscience and the Ann Romney Center for Neurologic Disease at Brigham and Women's Hospital. Dr. Jennings received his PhD in neurobiology from University College London, followed by postdoctoral fellowships at Harvard and MIT, where he studied the molecular and cellular mechanisms underlying embryonic development and the formation of neuromuscular synapses. In 1993, Dr. Jennings joined Nature as an editor. Shortly after, he was appointed as the founding chief editor for the newly announced at the time Nature Neuroscience. So today's episode, as you have deduced by now, is going to be quite different from what we've previously hosted on Stimulating Brains. However, Dr. Jennings' role right now, serving sort of as a connectome in Boston, is quite interesting and highly multidisciplinary. So we thought it would be great to hear his perspective on scientific publishing and to have him tell us a bit more about what it's like to have been an editor at Nature. So with that, I'll pass it over to Andy, who'll take over for the rest of the episode. And as always, I hope that you enjoy it as much as I did.
As you may know, to break the ice, I often ask about hobbies or things you might do beyond science.
Yeah.
Anything in your free time?
Yeah. Well, I like to run marathons, so I'm training for a marathon right now. I ran the Boston Marathon last year. I managed to qualify at my 10th attempt.
Oh, great.
And so now I'm running another marathon to try to requalify for next year. So that's a fairly significant commitment.
Great.
And
what does it take to qualify?
It depends on your age. So without disclosing my age, my qualifying time would now be 3 hours and 50 minutes. So
okay.
I think I can probably do that. If I don't get injured between now and next month, I should be able to do it.
Great.
So that's one activity. I like to climb mountains as well. So I try to do a mountain trip every year when I can.
Is it hiking or is it actual rock climbing?
I'm not a technical rock climber, but I like scrambling. We did, your European listeners may know the Italian Alta Via 4 across the Dolomites. So I did that last May. In fact, I did it before the huts were open, and so my friend and I were completely self-supported. So it was about a week of backpacking with no reprovisioning. So we were carrying a lot of weight over these wonderful mountain passes. It was absolutely fantastic.
That sounds fantastic.
Yeah,
I would love to see some pictures.
Oh yeah, well, I'd be glad to share. Yeah,
great.
Yeah,
amazing. So and then when we talk more about the professional life, so you have a slightly different career than most that I interview. But back when you were still involved in practical science, who were key mentors in the career or turning points for you along the way?
Well, certainly my thesis mentor, Martin Raff at University College London, that's where I did my PhD. He was really an inspirational figure for me. He is well known as author of Molecular Biology of the Cell, as a co-author of that very well-known textbook. Yep. And so he was an absolute encyclopedia of knowledge. He was also very well connected internationally, and so, we had a small lab at University College London, but he would bring in top scientists from the United States to meet with us and give seminars. So it was really a fantastic environment. It was international. It was my first exposure to international research.
Great.
And that stimulated my ambition to come to the United States as a postdoc. So that was really an important influence.
And you stayed ever since, right, in the States?
I did, yeah. So I came initially expecting that I would do a couple of years of postdoc and then go back to an academic career in the United Kingdom. But I ended up doing 6 years of postdoc, first at Harvard and then later at MIT. And I liked it and ended up staying. So yeah.
Where are you from in the UK, if I can?
So originally from Northampton,
okay,
which is known as also as the birthplace of Francis Crick. Discoverer of the double helix.
Yeah, of course. Yeah.
So
amazing. They just named an institute in London after him, right?
That's right.
Institute.
That's right. Yeah. I had no idea. Growing up in Northampton, I had no idea that Crick was from Northampton. He was not. It was just not known in the town at the time. It is now, and there's a big. I went back quite recently, and there's a big golden double helix in the main street in the centre of town. But there was nothing like that when I was a kid.
That's
so I didn't discover it until long after I'd left Northampton.
Yeah. Okay. So and then for your PhD, I think the Jennings et al. PNAS paper was in 1993, and we will only cover it briefly because it's a bit off topic here, but it described the muscle, and I also don't know too much about it, that's the other point, but it described the muscle-specific TRK-related receptor for the first time.
Yeah.
Is that correct?
Yeah.
Can you briefly summarize what it does, or why is that the receptor?
Yeah. So this was postdoctoral work at MIT in the lab of Steve Burton. We were interested in neuromuscular synapse formation.
Yep.
And as you know, the motor nerve grows out from the spinal cord and it makes contact with the muscle and induces the postsynaptic specializations that form a neuromuscular junction. And so we were trying to identify the signaling pathways that were involved in setting up the synapse, and we thought that maybe a receptor tyrosine kinase would be a likely candidate, and new techniques had just emerged for finding novel members of gene families such as tyrosine kinases. And so I was able to find this one, which we now know is a co-receptor for agrin, which is a key signaling molecule in neuromuscular synapses. And interestingly, it turned out long after I'd left the field that this molecule turns out to be an important target for autoimmune attack in some cases of myasthenia gravis.
Wow. Okay.
So the most common cases, the target is the acetylcholine receptor, but in I think about 10 or 15% of cases, it is this molecule MuSK. I mean, it was later.
Yeah, I know, of course, from clinical factors, now that you mentioned the MuSK receptor testing as well. And so antibody testing.
Yeah,
so,
and
didn't make that connection before. It makes sense.
Yeah, so apparently, there is now a clinical trial underway with a CAR T therapy targeted specifically to the B cells that are producing those antibodies. Amy Payne at University of Pennsylvania will actually be speaking at Brigham in a few weeks' time, so we'll get to hear the latest on that work.
Oh.
But I found it amazing that something that was originally discovered back in 1993, it's 30 years on that a clinical trial is underway. We don't yet know, of course, whether it will work, but optimistic. It looks very promising, apparently, from mouse models. So, but it's interesting how long it takes to get from a basic discovery to a clinical application.
Nonetheless, isn't that a prime example why we should do these basic science experiments? Because you never know when, I think another good example is optogenetics, where the original proteins were discovered long time ago, and without any, thought about using them later.
That's right.
And so, it's a good another example to show that if you hadn't discovered it, we wouldn't do this trial. No?
That's right. I mean, if I hadn't discovered it, somebody else would have discovered it. And if somebody else did discover it in parallel with us, we published first, but Regeneron also discovered the same molecule. And then they collaborated with Steve, my postdoc mentor, after I had left the lab, and were able to prove its function in neuromuscular development. So really, really nice set of papers, which I would have loved to have written myself, but that was the follow-on work from what I started.
And then, so if nobody did basic science research, then nobody would have discovered them. So if we wouldn't have enough funding for these types of
But basic science is the engine for
absolutely
clinical research.
Yeah. And then I think in that same year, 1993, you joined the scientific journal Nature as an editor. And while it has been a while, and I know you're not able, of course, to speak for the current editorial board at Nature, it's still so interesting to hear a bit back in the day. What did the typical day look like as a Nature editor?
Yes, it was a really, really fun job. I mean, I started as a junior editor at Nature itself, and I was given manuscripts in almost every area of biology. Okay. So we were really forced to become generalists. I mean, there was a lot of help from other more experienced editors, but I was thrown in the deep end and learned a lot about a very wide range of topics very quickly. And you're not expected, you couldn't possibly become a deep expert in all of these fields, sure. But you get a sense of, you get a really excellent overview of a broad field of research. You get to know who are the experts in that field. You get to meet them. You get to interact with them as referees and authors. And it's really, really a very stimulating, very, very, very enjoyable job. It was hard work. I mean, sometimes, obviously a journal like Nature gets an enormous number of submissions. A great majority of them are rejected without review, and part of the editor's job is to read through all these manuscripts and determine which ones are sufficiently promising that they should be sent out for external review. And so we would read large numbers of papers really quite quickly to make those determinations.
I think you mentioned recently that you had an hourly rate, or how quick did you get at
Yeah, when I was on a roll, I could do about an average of one every 20 minutes.
Okay.
Now, some of them you can look at, and in just a few minutes, it's obvious either that this is not Nature material, or it alternatively, sometime more rarely, but it's obvious that this is a very important paper that everybody is waiting for, and you can't not send that one for review. So there's a wide range, but the average will be 20 minutes.
Got it.
That's under pressure. I mean, nobody likes research that fast. Sure. Usually, if we could, we would spend more time on them. But when a big pile had accumulated after a trip, could grind through them.
Got it. And that would often really be reading the whole entire PDF or like paper at the time.
Yeah, well, back in those days, they came in FedEx envelopes on paper. Yeah. So it's a skim reading, and one of the skills that you develop is to figure out how deeply you need to read in order to get the gist of it. And some of them it's immediately obvious. Some of them you really need to read it, over and over again to get the point. Am I missing something? Is this, what is the author trying to say here?
It sounds great. And so the other point is I saw you did write editorials sometimes as well. There was one, The Death of a Synapse, if I remember correctly, that you wrote back.
Oh, that was in News and Views. Yes, that was for a paper by Rita Police-Gordon and Jeff Lichtman that was published. So it
was before your time at Nature? Or?
No, it was. I was an editor at Nature, and I was normally the News and Views are written by PIs, experts in the field, but since I'd just done a postdoc in neuromuscular development, the senior editors asked me if I'd like to write this editorial, write this News and Views. So I did, and that was a fun assignment. We came up with a very nice diagram, which Jeff Lichtman himself seemed very appreciative of this, and he used it quite often in his talks, and he was kind enough to acknowledge me as the author of this.
So great. So, I briefly looked at it, but the gist of it is you write about how the motor neurons would would select, and then also I think in the beginning many neurons would map to the same muscular fiber, but then some.
That's right. So you get activity-dependent competition and elimination of polyneuronal innovation. So as you say, initially one muscle fiber is innervated by many motor neurons, but they compete through an activity-dependent process that was just starting to be understood at that time, and signals from the neuron that gains strength then suppress the strength of the synapses that are losing out until you end up with just one motor neuron to each muscle. So if you didn't have that, it would your motor system would get very confused.
Of course, yeah.
So yeah,
makes sense. Do you still keep up with that literature?
I try, but I can't. I mean, I have so many other things to do.
Yeah.
You, and one of the things you have to do as a Nature editor is move away from your original area of specialization. Maybe not completely, but you have to, develop some objectivity. I think somebody who is just too deeply and narrowly attached to one, the area in which they do their own research is probably not. That's not the right mindset for an editor.
Great. So a new trend of many publishers that I perceive is to add numerous new titles. For instance, I think Nature just recently launched a subjournal, Nature Mental Health, but I think it's a trend that we see everywhere. Elsevier just created a journal entitled Deep Brain Simulation, while there's already a brain simulation journal, right, which is another subjournal there. So, and Frontiers might be the strongest proponent of really slicing up new journals with ever more focused niche targets. What do you think of this development? Is it a good thing or detrimental, or any thoughts on that?
Yeah, so the Nature stable has expanded enormously since I was there, and obviously, as you say, this is a broad trend in the field. I mean, partly it's just an inevitable result that the number of papers overall is increasing. I don't know what the current statistics are, but it used to be expanding at a rate of about 15% per year. I'm not sure if that's still true, but it's an enormous number of papers that need some home. So then, you can either have more journals with fewer papers, or you can have mega journals that just cover everything. I'm not sure that one is, it's not either or. I mean, you have both, and I'm hard pressed to say which is better or whether it's good or bad. I mean, the publishers obviously have a commercial motive, and they can. It's probably easier for them to charge more if they can slice it up into more and smaller journals.
Yeah.
But I think perhaps the identity of the journal in which something is published is not quite as important now as it was back when I was an editor. I mean, the way we consumed journals back then, certainly when I was a postdoc, I mean, we would go every week to the mailbox and pull out the latest issue of Nature or Science or Cell, and we would, browse through it in the T-ROM, go through the index on a printed product. Now, almost entirely driven by web-based searches, and then, you probably look at the journal as the identity of the journal as a stamp of approval. So if it's published in a more prestigious journal, it's on average likely to be more significant, perhaps. But that's not a terribly reliable predictor, as you all know. And you're probably going to search for papers in topics you're interested in, probably by authors that you follow, authors that you trust, and the journal in which it's published is less important.
Yeah, that's a good point. Yeah, because in 1989, you were appointed then as the founding editor-in-chief of back then a new title, which was Nature Neuroscience. Yeah, reading that is interesting to me, because in my life it always existed, right?
Yeah,
so I'm just a scientifically younger, academically younger than Nature Neuroscience. So at the time, Nature didn't seem to have so many subjournals. Did it? Or?
No, it did not. So it had launched Nature Genetics, and then Nature Medicine, and Nature Structural Biology. I think we were the fourth to be launched. Okay. So it was still relatively new. The others have been successful. I mean, Nature Genetics, the first, was impressively successful. I think it just caught this wave of the rise of genomics, and the Human Genome Project was getting underway. So neuroscience was seen as a potential next growth area.
Okay.
I think the concept of neuroscience as a field, it was not that old. I mean, it had historically been departments of physiology, departments of anatomy. So it was a subset of those areas, and it had become newly exciting, I think, as a result of the intersection of molecular biology and traditional neurophysiology and developmental biology. And so, the field had emerged, and I think there was an appetite for a new journal. I think in particular there was a need for a journal that would integrate all of neuroscience. So somewhere where people could read the most interesting papers in everything from the biophysics of ion channels all the way through to human cognitive neuroscience and functional imaging, and having a single forum where all of those different fields, they all could acknowledge that, for all the technical differences, they're all focused on this same fundamental question: how does the brain work?
Yeah.
And there wasn't any journal that really catered for that. And so we wanted to be that journal. I think we succeeded, and we made a very specific point of publishing papers in our first issues. And in fact, throughout my time as editor, I think they probably still do this. We organized the papers in a sort of sequence, if you like, a spectrum from small to large. So starting with biophysics and single cell physiology, upward to cell biology and systems neuroscience, and then ending with cognitive neuroscience and psychophysics. So that was quite a deliberate strategy. We wanted to send the message that this is a unified discipline, and Nature Neuroscience is a place where you can learn about the most exciting advances in any area of neuro.
That sounds great. Yeah. I mean, now that, as you say, people don't really have the book in print anymore that often, probably they don't even appreciate that. But back in the day, you read through the mailroom and see it. Yeah. So how was the role, except that, when establishing a new journal? Like what was probably you probably didn't handle so much yourself, but you would rather build it up and build a team, or how was that?
Yeah. So in the early days, it was all hands on deck, and I did my share of reading the papers themselves, but it was a much more managerial job. So it was Nature was a weekly journal, and there was a weekly rhythm of reading the papers, and then a few weeks later they would be published. The successful ones would be published. With Nature Neuroscience, we were building something from scratch. I think it was much more. It was sort of analogous to the transition from postdoc to PI. You're no longer just running your own research project. You're now in charge of building something over the long term, recruiting a team, training and mentoring people, building a reputation, building public visibility, building a network of connections and collaborators. So it was very analogous to that, I think. And I was very lucky. I got, we had a great team of editors, people, some very talented applicants who were excited about the mission and agreed to join us. So I was very lucky to work with.
Yeah. So, and it was highly successful. I think you did it 5 years, and it established the journal. So, what do you think makes such a successful journal? You mentioned good people as editors, but is there any recipe, or what is needed to?
Well, I think part of it was recognizing a gap in the market, is recognizing community demand. So in fact, we did a big survey. I was asked to organize this before Nature Publishing made the commitment to launch this journal, and to know, is the scientific community going to be supportive? And so I was given the job of answering that question and developing a survey and sending it out to, I think we sent it to several hundred prominent neuroscientists. Of course, I knew them by that time because I'd been an editor at Nature itself.
Yeah.
And the response came back overwhelmingly positive. So it was very clear that there would be enthusiasm, there would be support for the journal. So I think getting that right was key. I think credit to the other nature journals that had preceded us, so Nature Genetics and Nature Medicine, had already established themselves, and so we had a model that we could emulate, and we had the publishing infrastructure there. Obviously, the Nature name is tremendously helpful. Yeah. It sends a signal that this is going to be a competitive and prestigious journal, and then having the sustained support of the community, and I think having good editors who get to know authors and reviewers and gain their trust over time was absolutely critical.
Was that important, like the personal ties to authors or the personal, relationships with bigger lens?
Yeah, it is. I mean, people are not going to send you their best work unless they trust you to do a good job.
Yeah.
To give it a fair review. And similarly, reviewers are not going to agree to review. It's a lot of work to review a paper. They're not going to agree unless they feel that their effort is worthwhile. And then on the other hand, I think we needed to develop a sense of who's who in the community. Referees obviously don't always agree with each other. So an editor needs to, use judgment in terms of deciding whose advice are you going to trust? Who do I want to give an opinion on this paper? If referees disagree, which one am I going to believe? If they both make reasonable arguments, how are we going to resolve that? Judgments about how much work an author should be required to do to address the referee's comments. I mean, one frequent source of frustration from authors is that the competition for publication takes the form of just having to jump through more and more hoops. There's always one more experiment that you could ask for, and sure, it will probably make the paper better, but perhaps only incrementally better. And there's a cost to delay, and there's a value to getting what's already been achieved, getting that out as quickly as possible. And so I think one of the skills of an editor is to know when to bring things to a halt and make a decision one way or the other. Don't just sort of turn it into a war of attrition between the author and the reviewers.
It's very interesting. Yeah, makes a lot of sense. Because you often hear it can take another year once at Nature, right? If you have like a major revision.
Yeah, we tried very hard not to do that unless there was a strong reason. Sure. I mean, sometimes there is a good reason, right? Sometimes the authors are onto something really exciting, but the evidence is not yet conclusive. They have the ability to do it, but it's going to take some time. It's probably worth waiting.
Yeah.
I mean, one of the worst things you can do is contaminate the literature with, results that are inconclusive and turn out after the event, they turn out to be statistical artifact, based on inadequate sample size or whatever.
Of course, although I would say a lot of that is published, right? Just not at Nature.
Yeah, absolutely. I couldn't honestly claim that Nature never published. Sure. We never published papers that,
Of course not.
Yeah.
Makes sense.
Yeah, I mean, the literature is just full of, underpowered studies that it's very difficult to know what to believe.
Yeah, that makes a lot of sense.
Yeah.
Great. Yeah. So what do you think in general about the open access movement, the journals? Probably a good thing, but any thoughts on that?
Yeah, I do think it's a good thing. I certainly, I moved from Nature to Harvard and then to MIT, and I always had access. They have great libraries, so I had full access to literature. But I did a year as an independent consultant in between Harvard and MIT, and at that point I no longer had, as a private individual, I didn't have access to and you suddenly realize that, unless you're one of these wealthy institutions that has, a lot of institutional subscriptions, the average person, including researchers at less wealthy institutions, they don't have this stuff, even though it's, it should be public information. A lot of it was paid for with taxpayer money, of course. So I was very sympathetic to the idea of open access. I was very sympathetic to the idea of PubMed Central, which I think has been I think that's a really good thing. Publishers, of course, they were very resistant originally, and eventually they realized it was unstoppable. I think the public interest argument was overwhelming, and they didn't want to be on the wrong side of history, or they were the wrong side of Congress, or maybe more to the point. So I think that's been very beneficial.
Yeah.
As we know, some publishers have very large profit margins, and that's been widely discussed and debated. But so I think open access is great.
Yeah. Maybe to go a little bit into the prices, and I know you can't say so much about this because you've been an editor a long time ago, but I think the open access charges at some of the Nature journals, including also Nature Neuroscience now, rank at around $12,000. And I think you have a choice, right? You don't have to make it be open access.
Yeah.
But I'm sure this was different at the time, or it didn't even have that. But any thoughts on that development? Is it?
Yeah, well, the prices have gone up. I mean, I had not actually realized until you mentioned it earlier that they had risen so high. I mean, that really is very, very expensive. I mean, the publishers will say, and they will be right, that it has to be paid for somehow. That there is a cost. Sure. There's a cost to filtering. Editors have to be paid. Copy editors for the web, maintaining a website. It all costs money.
Yeah.
As if a subscription model no longer works, I mean, I'm guessing that they get very little money these days from private, individual subscriptions, and Google and et al. Have swallowed up the advertising revenues, so it has to come from somewhere.
Sure.
But I agree. $11,000 or whatever it is these days is a lot. I mean, one of the great things about PubMed Central is that you can see a near final version. You don't have to wait. You don't have to pay.
Yeah, that makes sense.
And that's comes with that. That's
and I guess one key difference for these Nature journals is that actually there are people that are hired, paying taxes, professional editors. So that sometimes I see as a real difference, because they have full-time professionals there, which is not the same in all.
Well, in academia, I mean, somebody is paying their salaries, right?
Yeah,
I mean, it's true. That nobody is working pro bono. They're being employed by universities or medical schools. They're being paid, maybe through indirect costs or whatever mechanism, but the money doesn't come out of nowhere.
Sure.
So,
no, but it doesn't have to be paid for by the publisher, right? If in many of the Elsevier journals, the editors might be scientists that do it near for free, or some do it for free.
Yeah.
On a lower editorial level, I think it's completely free often. So then they don't have to at least pay their salaries.
Yeah. And my only point is there is still a societal cost because
of course,
these people are drawing salaries and somebody's paying that salary.
That is true. Yeah, good point. Yeah. And then I think I just wanted to mention it because a few days ago, the entire editorial team of the Elsevier journal Neuroimage resigned over exceedingly high article processing charges of around $3,500. And I just thought to bring it up because it was a few days ago. And when the editors, I think, threatened before to resign toward Elsevier, the publisher declined to reduce the cost. So they really then did resign and now found a new journal with MIT Press called Imaging Neuroscience. And to me, this, also this Neuroimage moment received a lot of applause from the scientific community at Twitter. But it was also, I think, a testament that Elsevier did not want to, even when threatened, change anything in their policy.
Yeah.
Do you see chances of change when, to reduce the APCs, the article processing charges, when we do these things? Or do you think it's a futile struggle? And
yeah, that's an interesting question. I mean, I would say I haven't followed it closely enough to have a very informed opinion on that. But, the one thing that will make, that will cause change at places like Elsevier is large-scale reaction, such as what you're describing.
Yeah.
And Elsevier, from what I understand, has a higher profit margin than probably any other major science publisher. I believe it's higher than Springer Nature.
And any industry, I think.
So
yeah,
I don't know, but I wouldn't be surprised. Right up what is somewhere around 40%, probably. Right, it's very high.
Yeah.
So this will be fought out in the marketplace. And if MIT Press can serve that community in a more cost-effective way than Nature, if that's capitalism, I'm all for it.
Yeah, makes sense.
So, yeah.
Great. So as mentioned, you held the position of Nature Neuroscience editor-in-chief for 5 years, and that led to it being one of the preeminent journals in the field. And then what made you to leave that sector and go back into academia? I think in 2003 you first became. Let's briefly talk about that. Executive editor for all the Nature monthly research journals, responsible for managing the editorial department and developing long-term editorial and publishing strategies for the Nature publishing group. Can you briefly talk about that role a bit?
Yeah, so as the Nature family, the Nature Research Journal family had expanded, it needed more coordination.
Yeah.
And we wanted to make sure that there are uniform editorial policies and a uniform sort of public image, and there was a need for coordination. So I became the executive editor, so I had editorial oversight of all of the journals. So at one point I was managing an editorial department of almost 60 people. So it was a fairly big managerial job. I enjoyed it a lot. I was, became much more involved in the business strategy of the company, and thinking strategically about the scientific landscape and about where new publishing opportunities might arise. I was very much involved in the launch of the websites in the transition to online publishing. So when just before Nature Neuroscience was launched, Nature made what in retrospect was a very ill-judged decision to put Nature onto CD-ROM rather than creating a website. And I remember one of my colleagues at another competing publisher laughing at us for having done something so stupid. And in retrospect, they were correct. The CD-ROM was a complete failure. And but by the time Nature Neuroscience launched, we knew that we needed a full online presence. So I was very much involved in designing that website. We then we introduced what we called advanced online publication, which of course is just very standard these days. The idea that you would publish individual articles once they're ready before having to wait for the next monthly print issue. And so we developed all the sort of workflows and criteria for that, and the public communication about what that's all about. So that was really fun. But I liked the scientific aspect. I liked the hands-on editing and the decisions about editorial content of journal, and I like being close to the science and to the researchers. So I didn't really want a career on the business and financial side of publishing, and that would have been, the next step up the corporate ladder would have taken me further away from the research. So I was in 2004. I think I moved to Harvard, did a career change, become an academic administrator at the Harvard Stem Cell Institute. Yeah. So that turned out, that was describing our sort of baptism by fire in academic administration, because Harvard, as you know, is a very complicated place. There's the university, there's the medical school and the affiliated hospitals, and I really didn't understand anything about the political relationships between them. We were involved in stem cell research, which was at that point extremely visible public controversy. It was just at the time I joined Harvard, just at the time of the Bush-Kerry presidential election, in which human stem cell research became one of these sort of cultural wedge issues. And so we were very much in the spotlight at Harvard because we wanted to do, obviously, human embryonic stem cell research. So my boss, the co-director, Doug Melton, is a very prominent stem cell researcher. His kids, I had done a postdoc with Doug back at Harvard at a time when he was a pure developmental biologist. He was studying the early developmental, the control of development in frog embryos. That's what I worked on. Yeah. But then his kids were born and developed type 1 diabetes, and he switched his entire research program to understanding the development of the pancreas, with the goal of being able to regenerate or to grow neurons for pancreatic transplantation. And he became, a national advocate for stem cell research. So So it was so anyway, I was sort of thrown into that environment, and also at the same time, Larry Summers, the president of Harvard, was forced to resign over his controversial remarks over women in science. So it was an exciting 18 months, but it ultimately wasn't the right fit, and so I left after 18 months
to MIT, right?
And well, I did a stint as a private consultant, and then moved to MIT. So I was recruited. Yeah, go.
That was at the McGovern Institute.
That was at the McGovern Institute,
Technology program.
Directed by Bob Desimone, who I guess has been another very significant figure in my career. He's really a brilliant neuroscientist and a great science administrator as well, a really great leader. So I'd known Bob as a frequent author and reviewer at Nature, and he invited me to lunch, and said that he was looking for somebody for the newly launched McGovern Institute, and might I be interested? And I was ready to come back to institutional life. It seemed like a fantastic opportunity. I ended up staying there for almost 11 years.
Oh, okay, great.
So
I love the building, the white building, right?
Yes, that's right.
It's my I would say favorite employee employer in the world. Has been for a while. So it's such a nice building there at MIT. So you had a good time.
I had a great time. I mean, MIT is it's a fantastic place.
Yeah.
It's a wonderful environment. Really talented colleagues. And again, learned a great deal.
Yeah.
You mentioned optogenetics earlier, so I had a ringside view there. Got to know Ed Boyden very well. Ed is a faculty member at the McGovern Institute. In fact, Ed, the program that I was running, the technology program, we provided the funding for the first primate optogenetics experiment. So Ed collaborated with Bob Desimone, and they published a paper in Neuron showing optogenetics in monkeys.
Amazing.
So I forget what that was, 2007 perhaps, a little later, 2008 maybe.
Great. And let's jumping 11 years ahead, and now you are the executive director of the Ann Romney Center for Neurologic Diseases here in the hospital, and also the program for Interdisciplinary Neuroscience here at Brigham and Women's. Before we maybe talk about your role here, can you tell us a bit more about what the Ann Romney Center does, or what it stands for, what that means? Yeah.
So the Romney Center is focused on understanding brain disorders, and specifically neurodegenerative diseases as the primary theme. Ann Romney, of course, is the wife of Mitt Romney, prominent political figure, and Ann is a patient. She's treated here for multiple sclerosis. She's very public about that, and is an advocate for brain research as a result. She's a patient of Howard Weiner, who's the co-director. So the Romney Center was established many years ago by Dennis Selko and Howard Weiner. Dennis is a leading figure in Alzheimer's disease research. Howard is a leading expert on multiple sclerosis. They've been working together for many years, and the Romney Center has grown into what's now a large community of researchers, really united by focus on brain disorders, as I say, primarily neurodegenerative disease, so Alzheimer's, Parkinson's, multiple sclerosis, and other autoimmune and inflammatory diseases, but also brain tumors, so glioblastoma, and then gradually expanding to other fields as well.
Parkinson's as well.
I mentioned Parkinson's disease. I've said Parkinson's, other synucleinopathies. Yeah, I mean, so Dennis is primarily known for his work in Alzheimer's disease. He's been in that field for a long time. He's one of the most prominent figures in Alzheimer's. He also has a long-standing interest in Parkinson's disease, and that's become a major focus over the last few years.
Yeah.
I'd say one important research, been many important pieces of work, but Dennis, working with Tim Bartels, who's now at University College London. They were the first to look at the tetramerization of alpha-synuclein and to realize that the configuration of alpha-synuclein, its association or dissociation from the membrane, was an important contributor to the disease process. And so now that's led to a whole new field of work looking at the lipid composition of membranes, and that those are potentially druggable targets. And so Dennis and many other people here are working to pursue that and to try to develop drugs based on that concept.
Great. And in Alzheimer's, what is the main leading theme these days?
So there's, as we all know, I mean, the amyloid hypothesis has been a central feature of the Alzheimer's world for quite a few decades. Dennis certainly has been a leading contributor to that, and has been, I think, very much identified with the controversies around it. So the primary reason for the controversy, of course, is that many clinical trials have been attempted and an enormous amount of money has been spent, and they've been largely unsuccessful. The biological evidence, I think, is overwhelming. Right? The genetic evidence for that A-beta, the amyloid peptide, plays a causal role. I mean, there's very, very strong genetic evidence for that, and Dennis has contributed to that and to the cell biology and biochemistry of understanding the mechanisms behind those mutations. I think we've had, as I'm sure you know, as sure your listeners know, there's been this recent trial that's led to the approval of the first anti-amyloid antibody, lecanemab. And I think that's really a vindication of the work that Dennis and many other groups have been doing over a very long period. I mean, nobody's going to claim that lecanemab is the cure for Alzheimer's, but from what I understand from experts in the field, the trial was very well conducted and the results were clear cut. And there is improvement on the endpoints that seems incontrovertible. So that's the ultimate test of a mechanistic hypothesis is, can you show that it works in human patients? And finally, we have a clear-cut answer. It does work.
And I mean, we can maybe mention that, sit in the Hale Building for Transformative Medicine. And I think one idea of this building is that we have, everything from cell research, mouse research, animal research, but also see patients here in the building. Right? It has been designed with that concept in mind?
It was, yes. I mean, I came here soon after this building had been opened. And it's quite a unique environment. I mean, we have 3 floors of clinics down below and MRI down in the basement. And then we have, including the 70, yes. And then we have research, the wet lab research, including almost 3 floors worth of neurology research on the upper floors. So it's a really unique environment. I think what that means in practice is, anytime we go down to the cafeteria or we, come and go from work every day, we see patients and we see the people who are looking after the patients, the clinician, the clinical staff, and everybody else who makes the place tick. And it just reminds you every day, what this is about. What are the stakes? Why are we doing this in a way that doesn't happen in a pure university environment? It also means, it means many of the faculty here have joint activities. I mean, Dennis himself is both Dennis and Howard. They see patients and they run research labs. That's true of many people here. And it's difficult, I think, to run an intensive high-level research lab and see a lot of patients. But many people seem to, they will do 1 day a week in clinic, and that will keep them engaged, keep their skills sharp, and then they run their often very successful research labs in the other 80% time. I mean, I can only imagine how people do that. I don't have to run a lab and I don't have to treat patients, and
it is
impressed by.
Sure, it is. Me too, absolutely. And Is it possible to summarize Howard Weiner's activities in a few sentences? Probably not, but what does he stand for?
Yeah, so Howard is he's been a pioneering figure in the field of multiple sclerosis. I couldn't do justice to everything he's done, but he's really one of the leading figures in the demonstration that MS is a autoimmune disease and is amenable to treatment by drugs that target the immune system. And as I'm sure you know, I mean, many multiple sclerosis patients now do pretty well on some of those drugs.
Yes.
That's not true yet for Alzheimer's.
True.
So, it's a very interesting comparison. The MS world sort of offers you the hope of, this is what can happen once you get to the bottom of the causality of the disease, and you can start to develop therapies that target those fundamental mechanisms. That's where we're at with multiple sclerosis. It's not a completely solved problem, but it's at least partially a solved problem. Alzheimer's is not, but that's, we know where we need to go.
Yeah. No, and it's remarkable how different it is now. If I would get an MS diagnosis now, it's a much lighter thing than 20 years ago.
Than 20 years ago.
Yeah. So that is really remarkable. Cool. So what is your role in the center as an executive director? What do you do?
Yeah, I do a whole variety of different things. Yeah. So I oversee a large core facility. We call it the Neurotechnology Studio. It's a fancy name it's a fancy name for a collection of high-end instruments and expert staff who will help researchers to make full use of those instruments. So it includes microscopy, it includes genomics, high-end optical imaging. We have a 2-photon rig that's optimized for optogenetics and uncaging of for instance, we're studying synaptic interactions. So we raise philanthropic money to support that, and then we decide what kinds of equipment we want and what kind of support we need to help people make the best use of it. I do quite a lot of mentoring of junior researchers, so people will often come to me for advice on either manuscripts or grant proposals. So I read a lot of proposals and give advice. I go to a lot of talks. I tend to prioritize talks given by local people because I want to know what's happening within this community, not just within Brigham, but we're here within the Harvard Medical School environment, and within a half-mile radius of here is probably it's the biggest community, probably the world's largest community, one of the world's largest communities of biomedical researchers. Nobody can know everything that's going on, and most people, especially if they have to run a lab and treat patients, they just don't have time to meet everybody and know everything. So I see part of my role is to know who does what's happening around this community, and help make connections, spot collaborative opportunities. Somebody, will ask me, who can I need some advice on, MR compatible audio stimulation. Who should we ask? Or I want to get cerebrospinal fluid from a mouse brain. Who do I ask? And I know the answers to those questions.
That's great.
And many more besides. So part of my role is to help make those connections. I also get involved in big grant proposals. So proposals for new equipment, anything that requires coordination across departments or across multiple stakeholders. Maybe no one person is going to be willing to take the lead on behalf of a large group, so I can step in and help to coordinate that. So, for example, we just raised funds from State Funding Agency for a new brain scanner. This is a new 3 Tesla scanner with very high gradient performance, so it will be like a connectome scanner. Yeah, it's developed by GE. It's a head Magnus. Yeah. It's a head-only. It's a research-only instrument at this point. And that's coming to Brigham in September. It'll be one of just a handful worldwide, and one of the first scanners with this kind of capability that will be in a clinically oriented environment where we can really, it'll be in environment that is also used for clinical scans. This magnet will not be used for clinical scans, but it'll be in a bay adjacent to others. So we have all of the infrastructure to do research studies on patients in a way that can't happen in a typical university environment. So I think that's a real opportunity. I've been very much involved in that. I'm actually meeting with a team from GE that's visiting on Monday.
Okay, great. And yeah.
So the scanner is arriving probably in September, should be in September. So
great. Where will it be?
It'll be at 221 Longwood. Okay.
Yeah.
So the imaging center just opposite Harvard Medical School Quad. And then more recently, I've been involved in another proposal which has just been funded for a new mass spectrometry imaging system. And so the idea here is to be able to localize within sections of tissue, localize metabolites with high spatial precision. So, for example, you could take one application would be resected tumors, patients who maybe have been treated by chemotherapy, and then if they then need a resection, you can then look within the tumor and see where the drug is distributed and correlate that with the metabolic responses of the tissue to the drug. So, we imagine that drugs sort of uniformly bathe the brain or the body, because that's not true, and therefore distribution is much more complex than that, and is often critical to understanding why it worked or didn't work. So there's a lot of companies are very interested in mapping where do their drugs go at cellular or subcellular resolution, and we will be able to do that, as will be the first system of its type in the United States. So
it's amazing. Congratulations for getting both of these bigger.
Yeah, I mean, it's a team effort, but of course, I'm instrumental in pulling these things together. So for the mass spec, I worked with Natalie Agar, who's a leading expert on mass spec here at Brigham and Department of Neurosurgery. So she's very excited. I'm excited.
Sure.
We'll get this thing next year. And
congratulations. Really great. Yeah. So this sounds like, I think, the funnest part of your job description to be the connectome here in Boston, to know.
Yeah.
It's such a, when I arrived here, it was, I was overwhelmed how big it is, right? How many people there are just within Brigham, but then if you go to MD8, Martino Center, MIT, even, it's such a vast city of research. And I think Helen Mayberg once said that she usually, when she visits people at Harvard, she tells them what other people at Harvard do. So they might not.
I can believe it.
Yeah. So it's very interesting. So I want to be mindful of your time and want to wrap up with some maybe rapid fire questions. Sure. Yeah. Did you have any eureka moments in your scientific career, or also more editing or administrative career?
Eureka moments,
or just positive events. Anything counts.
Well, I remember how did I decide to go to Harvard? And I was a graduate student in London, and I had postdoc offers both from UC Berkeley and from Harvard, and I was just trying to, they both sounded great. I was weighing them both up in my mind, and I was playing in the university orchestra at the time, and I'm sitting there playing the violin, and I remember we were sort of sitting in this, I think I can remember it quite vividly. It was the Slumum of Shostakovich's Fifth Symphony. And I remember just sort of, in rehearsal, and I just suddenly decided, okay, I'm going to go to Harvard. And that was sort of interesting neuroscientifically, because, what happens in your brain?
Yeah, we got
life-changing decision in a context that was completely irrelevant, and there's nothing to connect Shostakovich to Harvard more than Berkeley, at least nothing that
there must be something
that involved the show.
Massive resonance.
Just the idea that, your life decisions are, stochastic events with ion channels opening, and who knows?
Yeah, sure.
Yeah, but
and maybe on the flip side, did you ever think this was a complete waste of my time, or this didn't help well?
Every job has components that are a waste of time. Carefully reading a paper that was later retracted, that would be an example of a waste of time.
Sure.
I've done that a few times. Won't mention names, but I go to a lot of. I try not to waste time. I'm sure. I mean, I do waste time. Everybody does, but I go to a lot of talks and I take careful notes, and that takes extra time. I take notes on the computer during the talk, and then I will often go back and read them and sort of clean them up a bit afterwards, just to make sure I can still understand them if I come back a few months later. Because I see a lot of people will go to talks and they don't take any notes at all, and either they have astonishingly good memories and remember everything they ever hear, or it's not a very efficient use of time, because a lot of it, I find if I don't take notes, most of it is gone. Most of it is gone a month later, and I have, a vague recollection that I was at a talk and it maybe the name is familiar, or some claim sounds dimly familiar, but I'm not going to remember it.
Yeah, that's a good point. I mean, so, the other question would be, how often do you actually look at your notes? Because that's sometimes what I realize. I often take pictures or, to take notes, but then I rarely revisit them.
I do, but that's become much easier now that you can search text.
Yeah.
So I keep them on my computer. I mean, when I took handwritten notes back in the day, I often didn't go back to them, but I do it much more now because it's so easy. And again, coming back to what we were saying earlier, I mean, if somebody asks me for advice on something, or they're looking for a collaborator, I will remember roughly, I maybe remember the name, and I can quickly go and find the talk and then search, just search the text of my notes for this particular point, and then give what's hopefully accurate information.
Yeah, that's great to say. What does the future of neuroscience look like in your opinion?
Well,
are we there yet?
Predictions are always difficult, especially when about the future, right? Yes. Who said that? Probably Yogi Berra. But it's what they say. I think it's Sidney Brenner said. It's new ideas, new results, new technologies in that order, right?
I see. Okay.
So technology development is really key.
Yeah.
I'm probably getting the quote slightly wrong, but new technologies are an absolute driving force. I mean, I was listening to a talk today over at Harvard Medical School, talking about RNA, single-cell RNA-seq and other methods being used to catalog the diversity of neurons, and they've just released a full atlas of the mouse brain. They now have what they think is very close to a full description of these cells. Yeah, it's just out on bioRxiv. It's not peer-reviewed yet. This is Evan McCusko's group, the Broad Institute. It's just remarkable, right? I mean, it's just been inconceivable. When I was a graduate student, and the technology has evolved so rapidly that these things can now be done, and it's just a full parts list or something very close to a full parts list. So, the next step is to map their connections, and again, that would have been incomprehensibly remote when I started out. And now here we are. It's within reach. We're not there yet, but it's clearly within reach with technologies that either exist now or can easily be foreseen. So, what's the next thing that we need to do? I mean, do higher resolution imaging of brain activity and in the human, non-invasive imaging of the human brain. Next technical advance in that will be huge. I don't know what it will be. Somebody smarter than me will have to answer that one. Understanding complex diseases, particularly psychiatric diseases, where we don't have monogenic causes. There's still not a clear pathology in the way that there is for something like Alzheimer's. I mean, Alzheimer's, is a great example, right? We've known about the risk genes for the highly penetrant risk genes like APP and the presenilins. We've known about those for several decades. We have very clear histopathology, and yet getting from one to the other, and then from that to the cognitive decline is been very, very challenging. Psychiatric disease is going to be even more difficult, right? Because we don't have the highly penetrant mutations, and we don't have yet a clear common pathological endpoint.
Yeah.
So understanding what goes wrong, and most importantly, what can we do about it once we understand it? How do we intervene? I mean, that's not to be, that's not a trivial thing. So that's gonna keep the next generation busy.
Yeah.
And say
that makes sense. I like that. Yeah. And then, do you have any advice for young researchers entering the field, neuroscience or academia in general? Lessons learned, or
study computation.
Okay.
So that's something that I wish I'd learned more about. If I could rewind the clock, I would have learned more math. I would have learned more computer science, and would have recognized the importance of statistics and quantitative evidence at an earlier point in my career. I mean, turned out I didn't really need it for what I've done, but if you're going to do research, you could never. Yeah, for a marathon, you can never have too much training. For a career in research, you can never know too much computation analysis. So that would be one thing. Changing fields, not getting stuck in one narrow field, I think is really valuable, because whatever you're doing, I mean, just as an advice to somebody just starting out, whatever you're doing now and studying now and interested in now, you will probably be 10, 20, 30 years from now, if you're going to continue in research, we'll be doing something different. So just develop the breadth of perspective and the habit of mind, the confidence to change fields and learn something new. I think that's a good intellectual habit to develop early in your career. It'll serve you well later on. Don't get stuck on, one narrow path that only leads to one place.
Great, yeah. And then missed opportunities in the field. So do you think we should be doing something differently that we, as a field, we're currently not doing, or should be doing?
Oh man, that's an interesting question.
Broad question, yeah.
Well,
anything potential?
I think one broad problem with translational neuroscience is the overemphasis on mouse models. I mean, it's sort of hugely tempting to work with, genetically homogeneous, well-behaved mice, but they're not a great model for the messiness and the complexity of human disease. To understand human disease, and particularly if we want to apply these new molecular tools, we need human brain tissue. Some of it can be done using stem cell models, but there's a lot that is not captured and will never be captured.
Yeah.
In a stem cell model, you have to study the human brain, and that's going to mean either post-mortem material or new non-invasive ways. I mean, we can imagine, taking, and this is one thing that I think may be promising for the future, is the ability to, I mean, you will, as a clinician, you can judge the plausibility of this, but the possibility of taking very, very small biopsies from the brain that are so close to non-invasive as to be completely harmless. There may be opportunities during surgery to do this. And, we can now get an enormous amount with modern molecular techniques. You get an enormous amount of information from tiny quantities of tissues. So you don't need to take, cubic centimeters of brain or to grind it up and analyze. You do it with, 1,000-fold less or even less than that.
Yeah.
So I think that's going to present new opportunities.
It's interesting. There is the idea in the field currently that during the brain stimulation surgery, you insert microelectrodes sometimes and then pull them out again and put the actual electrodes in. Yeah. While pulling them out, you will have some tissue. Yeah, that can be analyzed. And
So we need to bring, the people who do that, we need to bring them together with the people who have the molecular techniques. And importantly, not just molecular, but spatial. I mean, this is one of the new waves of biology at the moment, is the ability to combine very highly multiplex molecular methods with spatial imaging. And so we can say not only, which 1,000 genes are expressed here, but exactly where at the cellular and subcellular level. that I think is a really powerful combination, and we're just starting to see the fruits of that.
Yeah, great. Okay. It's interesting to hear that from you, because especially what I'm thinking of course, basic animal research is very important, no doubt about it, but there are also in many fields, especially in the brain, there haven't been so many wins yet, right? Where led to actual translation in new therapies. And so I really very much agree that human research is very important, and but it's of course very challenging, especially if it's wet science. But with epilepsy surgeries, there's another way to get to tissue and these things.
Yeah, it'd be great to think more about how we could, if Brigham can't do it, who can?
Yeah,
right?
true.
We should be well positioned to
absolutely
make this happen. So it'd be fun to brainstorm.
Great. Is there anything that we didn't cover that I know I've taken a long time of you, but any question you would have loved to have covered?
No, I didn't think of any. I mean, I think we've covered a lot of ground here.
So then, and thank you again, Charles.
Yeah, no, it's a pleasure. So I hope this will come out well. I look forward to hearing it.